software center. I can see this, the latest the 2025A version. I believe that's a, there even, so let's install this, the better version, the latest. So this will take some time, so we just let it go. So you can use your Tamu email, because every one of you should have a license already. So 2025B version, log in with your student Tamu email address. Mm-hmm. You can use that, but it's okay. It's slower, but this one will install locally on your machine, so you have more control. Yeah. You can also use your own laptop. In previous years, some students use their own laptop. But I just let you know, you have license if you use your student email. This license includes MATLAB and many toolbox. For example, the statistics and machine learning toolbox. If you use your personal ID email address, you won't have access to those licensed toolboxes. Will be essential, some of them for us, but take advantage of your student email account. So this will take some time to set up. So that's a place where I said there are many associated toolbox which you have access using your license, student license. I don't know how many show up on your screen, but I have the full list, but I need MATLAB itself. We are not doing 5G, so we don't need 5G, we don't need audio, we don't need automated driving. That's for other people. We need bioinformatics, and I will show you which one we need. You can always, if you miss one, you can always come back later to add that in. So it doesn't really matter at this moment, but I just let you to check several essential ones to make sure we can run what we need today. We need curve fitting, okay? We will need global optimization. We will need image processing. We will need optimization, parallel computing. You know, you can select them all, but it will take a longer time to download and install. Many of these functions we really don't need. So that's why you can browse down. Make sure this will be checked, okay? This is a statistics and machine learning toolbox. We also, for the language model, sometimes we also need this text analytic toolbox. Okay, don't worry too much if you cannot follow me to check exactly toolboxes we need. But today, make sure you have this statistics and machine learning toolbox checked. Okay, otherwise, the rest of them not essential for today. Mm-hmm.
So how do I check whether the required
It's already in there.
Yes, yes.
You can check add-ons and then see whether manage add-ons,
Okay.
and then you can search. It's still waiting, so you can search whether you have the particular toolbox installed or not.
Okay.
Mm-hmm.
That would be the bioinformatics toolbox and the statistics toolbox.
Yeah. Mm-hmm.
Just those two?
These two will be enough for today. Yeah. Mm-hmm. Yeah. So today we are going to do this MATLAB 101 first. Okay. So you get familiar with the interface and what you can do, very basic. Then we will jump to 10x genomics cloud. So in this place, you will learn how to run Cell Ranger. Cell Ranger is an essential tool once you have the FastQ files, FastQ. And you put into Cell Ranger, so Cell Ranger give you the expression matrix. So for your own project, you send the sample to sequencing center, you get FastQ file back. And you can just upload to this cloud to run Cell Ranger, you will get expression matrix file. And once we have this, I will show you, we are going to come back in MATLAB to run SCGEA2. So this is an application called single cell gene expression analysis tool. So we are going to put this matrix into this tool and to start to do the analysis. So that's what we are going to do today. Okay? Okay, because I have already successfully installed this, I have this interface with my account. Okay, so I just want to make sure you have this. I will go around and make sure everybody on the same page. So this is a MATLAB workspace. You can have a file structure, and that's your C drive under your documents. You can put files, folders inside here. This is the workspace. It's a memory, and what's in your system. The variables will be here. And that's the place where you will communicate with this interface. If you haven't ever used MATLAB before, the best resource for you getting started is this link. Okay, if you click, there's a whole set of these tutorials and make your start very easy. This is a commercialized package, so very well designed. Okay, so it's an industrial level environment. So many of you have this R or other Python background, so it's very similar. So all the computer languages are similar to each other. You can use this to do this simple communications. If you do 1 plus 2, you get a 3. So this is the way you communicate with this, and you can call functions if you do a list of random number. See, I will need 10 random number, 5, okay? And give this random number to a list, so this will give you 5 random numbers. Want to make this bigger. Okay. So I suggest you click this and to take a look what's in there, and I will go around and make sure you have the thing you need. Still waiting. Okay, let's wait. Need time. Over there you have, okay, you're almost there. Okay. Still, yeah. To make sure everybody. Where you finish install MATLAB? Yes. Okay. It should be, yeah, so if you do this already there, yeah. You need to log in as well, yeah. So the first tutorial here is the desktop basics. So it will show you this is menu, here is toolbars, different areas of this workspace. Say I create a variable called A using a function. This function generates 5 random numbers between 1 and 0 and gives the values into the A. So that's why I can see there's a variable named A, this small a. And the value in there is this, and the size, the dimension, 1 by 5. In MATLAB, the variable names are case sensitive, so lower a and capital A will be different. Okay, normally, as a convention, we use small a to hold integer, single numbers, so scalar value. Sometimes like vector here, and use a big A to hold the matrix. Okay, if you have a say 5 by 6 of matrix, and you put a big A just for yourself to remember. I will show you some function which you can create arrays. Arrays are, this is an array. So there's one way you create an array, which is 1 by n or n by 1 vector. So that's a list of numbers held in a 1 by n or n by 1. So matrix will be n by m, so 5 by 4, 1,000 by 1 million, so it's a matrix. So I show you how to type those in and use function, for example, to create those. So those are the very basics. And if you have R background, you know you can do the same thing. Okay. So if I run this function again, it's a random number, so next time you will have not the same number. Okay? And you can see to use this function, RAND, which is a built-in function, you need 2 numbers. Here I put 1 and 5. Inside need 5 array or a vector A is 1 by 5. If I change this 5 to 1, I will get a vertical vector. Change this to 4, then you get a matrix. Okay, as I said, the better to put capital variable name to represent a vector, a matrix. Okay, so far everything is smooth, right? Okay. So I'm going to create a matrix by just type that information, to type those in. So what I can do, I'm going to create a matrix called a B. I don't know if you can see here. I make a font size bigger. I'm going to type 5 by 4, same dimension, same size matrix as A. I'm going to put some numbers. I just make it up. So I know the first row I will have 4 numbers. So I will put say 23, comma, and 12, and then put a 0, 3. So this is my first row. And then put a semicolon, so to tell the program to know, okay, I'm going to type the second row. So semicolon to change the second row. I don't know in R is the same. Maybe. Okay, so 1, 2, 49, 0. So how many rows I already make up? So this is 1, first row, 2, 3. So I need another 2 rows in order to make it happen. So I need, it's too long, so I put 3 dots here so I can continue. 56. 0. Another 4 numbers. 6, 7. Okay. I need one more, because I want to create a 5 by 4 matrix. So now I have a 1, 2, 3, 4 rows. I need one more. Okay. Okay, so you can, if you don't want to type this comma here, and just put a space, so the program should know. All right, I manually create a matrix. So if you miss a 1 of 0, you will get error message because you cannot miss anything, right? If you forgot to put 1 number here, there's some missing information, you will get error message. But I was so careful to make sure every row is 4 numbers, and I type them in the 4 elements, not missing any one. So, but at the end, I come up with this. So those are the random integers. I just pop up. I don't know from my brain, and I just put a random number there. I have no any models in my mind. So if I want to type in a series numbers of a human height by centimeters, I probably will type 160, 170, something like that. But here I just put some random number, which randomly generated from my mind. With this, you can see, I want to extract number this position. So this is row 2 and value 3. So if I type 2 and 3, so you see this is a row and a column. So this will extract 49 value. So this tell me in the B matrix at position 2 and 3 is 49. To get the whole row instead of one value, so I can do this column symbol to extract the second row. Okay. Same thing, you can extract the whole column. So this will be the column number 3. Okay. So this is column number 3. Okay, you see, when I type this B in, I use a squared bracket. But here I use this normal round bracket in order to get the index, extract the information. But once you want to construct a matrix, you use this bracket, squared bracket. So I will create a squared bracket inside the index, see 3 and 4. So this will extract third and fourth column from that matrix. You will get familiar with this difference in the different brackets. So this squared to create a matrix, to create a vector. These are the index and give to this index operation that will extract columns 3 and 4 out of this B matrix. Okay? A lot of things with, it doesn't have to be continuous. So 3, 4 is concatenated, continuous, but you can jump from 1, 4. Same thing if you want to do the rows. Change this to here. That will be extract row 2 and 4. Okay, from that matrix. So if you do 6, that will give you error message because you don't have row 6 in your matrix. It's okay. So this is our B if we do B plus 1. So this operation will increase one value for each element inside the matrix. Okay. So this operation is on the matrix level. So this plus 1 gives every element inside there a 1, increased by 1. And the result will be saved into C. If you pay attention to here, we have A is a 5 by 4 matrix created using a function called random. Then the B is we type that in manually with a squared bracket. And the C is derived from the B by adding 1 value into there. You can do multiplications by, say, 2, okay? So this operation, multiplication with a whole matrix with one scalar, one single number, will multiply every element inside here, okay? When we talk about the matrix to matrix multiplication, that will be a slightly different thing, but we are not going to talk today. So if you cannot do B times A, because A is here, B is here, you can do A plus B, B plus A, but you cannot do A times B. So if you remember, we have A here, we have a B here, and A plus B is every element inside A will add up with a B, but we cannot do A times B. So A is a random number matrix with the values between 0 and 1. And the B is a matrix contains only integers, some zeros as well. So if I do this A plus B, I introduce some kind of a noise, random noise, into the B matrix. This noise is uniformly distributed noise level between 1 and 0. So that's the results. Okay. I think in the last couple minutes, I just go through. I just have gone through this. Very quickly covered what the interface looks like, how you create vector or matrix, how to do this indexing, how to extract values by rows, by column, by individual numbers from a matrix. And then workspace variables. So we very quickly cover those. I think I'm going to jump to characters and the text. This will be defined as strings. We will use this to hold the gene names, okay? Then we can visit this page and see how we can call a function. We already call a function in the random generator, okay? So let's start to define the strings. So if you gave a name variable name the G1. So this will be a double quoter, and then this, for example, AHR. So this is one of the gene name. So unlike the integers or other numbers, now we put a string inside this G1 variable. We can have a G2, TP53. So the second gene, we can define a list, G list. I can put a G1 here, G2 here. Okay, you may have some idea what I'm doing here. Remember, I type integers. I have a 53 semicolon 1. In that case, I create numeric variable vector 51 and 1, 53, 1, this vector. Instead of doing integers, I put strings G1, G2 in there, and that will create a vector contains 2 gene names by rows. Okay, you can increase this, and if you have more genes, you can just add up, create your list of genes. So in this operation, I will take gene list itself, then put one more gene next to it. So that will increase the length of the gene list by 1. So how can I extract numbers in there? So I will extract number 3. Number 2, this will be TP53, right? So the same kind of the array indexing. So I just add more genes in there. So this will be our gene list. And this B will be our expression matrix. So let me replace this into another name. So in this case, I just rename the B into a variable named big X. Okay, I can rename gene list into a G. Simplify this. Okay, this will be our very basic data structure we will play with in the real data analysis. We need 2 things. One is a matrix named variable X. It contains genes and cells. So in this case, will be 5 genes and 4 cells. The last gene has no expression in all the cells, so all zeros. So each gene have its name, so that name will be given into the G. So big X, small g, just remember, that's what we always need for the downstream analysis. So we will process data, go through cell ranger using real data example. We will get a gene expression matrix, the files. This will create X. What is X? It's expression matrix and a G, a list of genes. So these two things will be what we need to give to this tool to do downstream. Okay? This big X will pretty much look like this. There are a lot of zeros, sparse, and some cell express this gene in low level, and suddenly there's a cell express this gene really high level. So this is what you will see in the real data. So that's this part, and calling a function. Examples. You define an array and give a name A, and this is a function to get the largest number out of this array. Then you have a B, another array, and you want to union, and you found so 3 are the shared, they merged. Okay, so this is a kind of operation. So you give the max A a number, which give this bonds, found the largest and the smallest, and you display array, a string. So those are the functions. You call functions to achieve something. It's very basic functions. So this function will clean up your this command line dialog window, CLC. Okay. So we can see which rand. Remember, I use random function to create a list of random numbers. But I need to know where this random function is located. So if I do which, you will see, okay, this is MATLAB itself. There's a rand function in rand, okay, inside here. There's another rand function, which if you add n, is also inside here, this folder. So this random function generate random number between 0 and 1, uniformly distributed. This will be normal distribution. So if you do, that's around 0. So if you do n1 and 5, you get which numbers is centered in 0, centered at 0, and then there's bigger than 0, lower than this normal distribution. Okay. So this which will tell you, okay, where your function was originally located, so you know what it is. if you do which PCA, you will see this function is inside a toolbox statistics, okay? It's not a built-in function, it's implemented in the toolbox. If you want to learn more about these functions, then you do doc, and this like help documentation to show you, okay, this is a random n function. It's normally distributed random number function. And how you're going to use it, so different ways there. Detailed documentation. So you can do this. This will create 5x5 matrix of the random number. Okay, any questions? Easy, right? It's okay. Let's see what we can do. I think some plotting function will be interesting. So 2D, 3D. Yeah, if you copy this, then you can do 3D. So you get X, Y, Z, and then you surf. So just you can type that in if you want, but you get a plot like this. Okay, so this is the interface for the plotting, and this is some MATLAB code to generate X, Y, Z coordinates, and you plot them out using the surface function. So this one is interactive figure. So if you move your mouse cursor to here, you will see some small hidden two sets. So if you move your cursor here, you get this. If you click this highlight, it will allow you to rotate this figure. Okay, so there zoom in function, home, and this hand to push to drag figures, add data points. So there some hidden tool here, and also there are many functions above this figure. You can add toolbars by just click this, and there's two bars here. You can copy figure here, and you just click this and paste into the Word. Okay. All right, how about we have a 5 minutes break? Okay. Okay, we are done with this part, and let's move to Cell Ranger. We will come back to MATLAB later. Okay, clean up this. So in Teams, I provide a link. So this cloud service is essential because Cell Ranger only run on the Linux machine. It's not so convenient for us if we do the command line. We can do that, but this gave us an interface which we can just drag our files in there and then let the cloud to do the job for us. We need to sign up. It may take some time for you to log in, and any Gmail account should be okay. I also prepared a couple of this FastQ example file. Files, not too big, so you can download onto your local machine, and we are going to upload those files to the cloud to mimic exactly what you will get from your own data. This is only one sample, okay? One sample associated with 4 files, FastQ. So I just download. So how can you tell this is one sample? If you look at the label of this, this is customer self-defined. So this is some kind of a control vehicle at A2, and the rest of them will be length of sequencing length, and R1, R2 are the pairs, pairwise. They're index 1, index 2. So depending on the version of your 10x genomic systems, current version maybe you only need this R1, R2. This is like some previous version. You also need index 1, index 2. For this example, we need 4 files, but your new generation of the data generated from the new reagent may only require two pairs. Right, so I download all of them to my local machine. So I'm going to upload this set, the whole set of the 4 FastQ files for one sample into the 10x Genomics cloud. The cloud system will utilize the label to group them together. It's smart enough. It knows these 4 files belongs to a whole set. Make sure you know where you downloaded the file. It should be inside your downloads folder. So I can see what I download is stored in my downloads folder. There's 4 different FastQ files. R2 and is the largest. So come back to here, and you may see there's your account setting profiles, billing information. The service is free, but if you want to long-term store your results, you need to pay for the storage fee. So what we need to do is so this is for spatial transcriptome, looks like, in situ. And this is for the setup of the instruments. So we will go to the cloud analysis, and we will set up an example project, and then we upload files to that project. So let's go to the cloud analysis. What you will do is to click this blue button to create a new project. I already done this. I create VIBS 6045 as an example project. If you're from Dr. Shetty's lab, you can see some of my project for Dr. Shetty. But anyway, you come to inside here. So I'm going to delete what I already uploaded. So that I will have exactly the same interface as what you will see on your screen. So you create a project so you know what is the for. Remember to make some so it's easy to forget what you're uploaded, what your sample is. So let's see, I will look at, check whether you are on the page ready. Are you okay? You have on this page? Okay. You guys are really quick. I can't believe you can easily follow those steps. So if you know where your 4 FastQ file is saved, maybe you select them and drag them, put into here. So this upload manager will start to upload those 4 files. and using the tag information to group them into one sample, okay? So in my downloads, I select them all, and then start to upload. Hope we are not compete for the same bandwidth. It shouldn't take too long, but in reality, most of the files will be bigger than this. This is only 1.5 gig. A lot of deep sequenced sample will have much larger file size. So you should get the information from your sequencing center, which reagent you use, which version you use to prepare this sequencing library. Whether it's a 3' 5', so some kind of those specification, you need to have those parameters. You should know those knowledge information before you run next step. You also need to know your sample is from which species. Is a human sample, mouse, or something else. It's not uncommon to have a human and a mouse sample together. So there are some like a humanized mouse sample. So you inject human cell into the mouse. Okay, you collect, dissect the whole thing, and you have a mixed human and mouse. There are options you will see. You need those information. So those are very basic information, right? So my upload manager said, oh, I complete the 4 samples. And you will see automatically this has been given a name, because all the files start with this name. And this is like a same flow cell ID. So these 4 files have been grouped automatically. If you have another 4 samples, it will group at another sample. All right? Oh, job file here.
So what I've done is, I was trying to put this, but then it says, make sure it follows standard naming
Yeah, so
conventions.
yeah. So where the file you download maybe not complete. Yeah. This is not.
So it is not complete.
No, this is not zipped. If you go here, yeah, make sure you upload a zipped file instead.
Okay.
Yeah, so there's another issue I should warn you. You should know in Windows system what the file extension is. We need a GZ. That extension will be hide by, because as a non-extension file, so you don't see that. So we need to know if a file, so this file, end up with txt or something else, file end up with MD. So we need information about this extension. The file you download will end with GZ, which is a zipped file. So if you click that in, then you see FastQ in there. You cannot upload things inside the GZ. You need to upload the whole zipped file. So that's the problem you had. Make sure those operations will be necessary to make them correct. So now we can select the whole set. Remember, this is a set of FastQ files. And here is the information you need to know when you prepare your sequencing library. Which version of those you used. For this sample, let's select universal 3. Okay, click that inside 3 primer. Gene expression. Okay, so this gave the cloud the information, which version, chemical we used, what direction, the 3 primer and 5 primer. This all should be set up for the experiment. So now we're ready. So now you select this and you create new analysis. So now you have FastQ file uploaded. The analysis we will do is to give those files to Cell Ranger. There are different versions of Cell Ranger. The simplest one is to get the expression matrix. Okay, so this is straightforward. So this is our FastQ file set with 4 samples. If you have a different sets, then you can select a different one, but ours is just number 1. And you will select the pipeline. So if you look at this, because you only upload one site, there's only one available for you to choose from. And here you have a different version of the we just need the count. Okay. So select this count, type some here is a reference. This is important. So it's human sample or mouse sample or the mixed or something else. If you have zebrafish sample, you can do it, and you need to do a customized reference. Reference means the transcriptome information you provide to the system to allow this mapping onto that system. Here the cloud only provide human and mouse reference. So this sample actually is red. All right. We do have a red version. So click red, and the rest of them just optional. So you don't need a BAM file until you need to have those splicing information later. So let's just check off this. And then you can click run. Okay, so we upload the files, select the parameters and the reference, and run this. We are waiting for this matrix to be generated. Are we on the same page? You also get something like this. We don't have to wait until it's finished. We can keep going and use other example data to show you next step. Okay, let it run, and it may take 10 minutes. Okay, this small file. So now we can leave this. You can shut it down if you like, because this cloud, you can log in later to retrieve the results. You can leave the window like that. Let's come back to MATLAB here. We are going to install this tool so that we have something to use to analyze this data. Okay. To do that, you click this add-ons and explore add-ons to search for this particular toolbox. There are many toolboxes you can select for. Some already included in your license, for example, internal toolbox. So if you design this, you can use by click. Well, here you have to do is just search GEA toolbox. This is the only toolbox which is designed for single-cell data analysis in MATLAB. So on this page, you can just click Add. So once you install the toolbox, you can type in this SCGA2. So that's the only command line you need to use. Basically, remember it's a case sensitive. Everything is lowercase. So you can bring up this interface. Okay, I want to make sure everybody have this set up. Not yet. So install, you can close this. You only need this particular interface. Yeah, type that in, should be, yeah. Yeah, so you have it here. Yeah, you can go ahead and click and then see what's import data. So this is still working, right? Create analysis. Oh, you select the library. Yes, universal G expression. Should be this one. Yeah. Oh, you need to select this so it knows. Yeah. This whatever you can type in, then this select red. Yeah, red. No, that's human. Yeah. Uncheck this. Take too long. Yeah. You need a name. Good. Yeah. Yeah. So you can use example data to play with this. So that will be the interface. We will import data which generated from a cell ranger using this to get this information in. for some reason, I cannot get this particular file run correctly. Maybe it's a mismatch of those reagent version. So we can leave it like this, but in real applications, you should be able to easily generate your own data. So what I did, I provide an example data already in the download folder. So this is called, if you found a file, which this matrix file will be called H5. So something in front, filtered gene expression, and you will find H5 extension for this file. That's the file you will need to use if you go back to this teaching here. This file, okay? Let's download. So Cell Ranger will export a set of standard H5 file. And this is filtered feature. Feature means gene expression, gene, and the matrix H5. This file, the download, and you know it's in the download folder, okay? If you don't have this, refresh your web page. You should have this. I just uploaded them. Now come back from MATLAB to the MATLAB. Here, we can import data, and you can find this particular option, which is the first option under this separator bar. This is called a 10x genomics H5 file. It's the file it's generated from Cell Ranger. Contains gene expression, matrix, and the gene name. So you select this and find the one you download in the downloads. And read that in. The first dialog you will get is something like this. This information is because the list of gene, there duplicates. Some gene called TP553, there another gene called the same name because of mapping issues. So we need to have a solution. There are different ways to solve this problem. If you don't know through the pipeline, you don't know which option you select, just click the by default, okay? There are different ways you can just rename them. You can merge the average, the gene expression for the same gene name. You can delete. So there are different ways to do this. If you don't know which one, you just click method 1. By default, this pipeline should give you the most applicable options. So this data set contains 36,000 genes and 2,390 cells. So you can imagine this is a big spreadsheet with 36,000 rows and 2,000. The number of cells is not large, but the number of genes, that's all the red. This example is from a mouse. Okay, all the mouse genes in there. So these dots, each dot is a cell. So if you count, it should have 2,390 cells. in there. So this figure, as you know, there's small toolbars there. There are buttons you can click to rotate, to save the picture, to zoom in, zoom out. They're all there by default. Okay, so that's what I'm going to rotate, look at the different angles. Okay, so that's your, I think that's most we need to do today, is to know MATLAB, know how to get the data in where you have real data, what to put, how to get this and into this interface. Okay. So for the next, you will check the first button, which is the second row here. Here you will have a list of genes. You can select to look at, okay, those are the genes. The gene names, not complete. You will be able to see the expression level of some genes in those cells. Okay, so this is a very basic exploration tool. You select one of the genes and look at their expression among these cells. If you like a dark mode, you click this button here. There's a switch. Okay. So this data set is not annotated yet, not processed. If you want to take a look of this processed data, you can come back to this page and load example data. Here you select and you load a processed data. So in this case, you will have an annotated data set for you to play with. There are many buttons you can feel free to click. For example, these add labels, those buttons you can check and play with. It doesn't matter, it's all in the MATLAB environment. If it's crashed, you just restart. Yeah. So explore some of those functions if you like. So in the whole semester, we will study a lot of those functions, underlying principles, and how the algorithm is going, how this unprocessed data will be clean, will be clustered, annotated. If you have two samples, how they compared, how the network will be inferred, how the differential expression, differential variability, all the analysis will be conducted. They all are in the manuals. Some of those commonly used will be given as buttons, but the principle behind will be given during the lectures. Okay. So we still have some time, I believe. Let me know if you have any questions. So we start from very simple ones. I'm glad that then you can catch up very quickly. all the computer system has been set up easily, smooth today. I expected some problems, some issues, but seems okay today. In the previous years, sometimes one of the computer not working, student have to shift to another computer. But today everything seems very smoothly ongoing now. Yeah, I encourage you to continue to explore the resource provided by MATLAB, getting started, familiar yourself with those. They are interactive learning, and this is very useful, okay, to lead you. There are many, many videos to show you the capacity of this working in a development environment. So we will be using this interface a lot, but in the same time, I will show you once you click one of the buttons, what will happen if you don't have those buttons, and what's the command line behind each of this, okay? Yeah. The name of the cells, so that's annotation. So yeah, you still have a couple of lecture away from the, yeah. Oh, you want to see this label here, this label, yeah. Cell type. Yeah, that's okay. That's a question. It's already annotated. Yeah. Got this. Okay, this is a example data. So you go back to here to load example data. Click OK, process. If you roll, then it will be not annotated. And this is annotated. Oh, nothing here. Yeah. Maybe you zoom in too much. I don't know. Thank you. Yeah. Question? No question. Good. Oh, okay. Very cool. So this interface is working product. So you can use it to do the real analysis and to generate publishable results. I have a lot of collaborators using this. They extract the screenshot and they can publish in the journals. No problem. Yeah, you have some issues here. Yeah, so you continue. Only this. Not installed. So have you installed that? Get it. Then you search for SCGEA. Yes, yeah. You install it. Yeah, so you need to log in. After you installed, then you should have that. Yeah, okay. Oh, good. You like to use black Net mode. So another way to get the data into here is to download the published data. You can use geo accession number. So give the number of the sample, then you should be able to extract the information from the geo database directly. There are some examples. For instance, this number here, as an example, if you found it in geo database, is this particular sample. You see the sample ID, okay? GSM end with 68. Then you see this H5 file is here. So it's downloadable. So this data should be able to, if you give this ID here, it should be able to go there and download and process. So I'm showing you to download the example data from the geo database and then automatically process it, including data cleaning, cluster annotation, so you can get the information, what the cell types in that particular cell sample. So this is our annotation, automatic annotation for this sample, okay, by using the sample ID. Make sure you know, if you give an ID here, make sure in this record there are files in there. If it's empty, the author didn't upload the correct format of files, you don't be able to import into the tools, okay? We need the information to play with. So this sample contains neurons and fibroblasts. There's some misannotated cell type, haplotype. Then you can come back to this. This is neuron stem cells, maybe differentiated cell culture. So some has been annotated as a fibroblast, but there are many neurons. Okay, so this is the computer session for this week, and we'll see you next Tuesday, and we'll discuss matrix, okay? Yeah. Today we stopped earlier than expected because I gave some buffer time for the issues, technical issues. Since we don't have technical issues today, it's really good. So next time, if you sit in the same computer in front, you should already have MATLAB installed, or the toolbox is already there, so it will be much quicker to get into the real analysis.